Welcome back to 8 Square Edu. You're watching the series on molecular phylogenetics, and in this we are going to talk about tree topology and types of phylogenetic trees. Forms of tree representation. The topology of branches in a tree defines the relationships between the taxa. In each of these tree representations, the branches of a tree can freely rotate without changing the relationships among the taxa. So there are two forms of tree representation, cladogram and phylogram. Cladogram. Cladogram is a branching diagram showing the relationships among a group of clades. The shape of the cladogram shows the relatedness among a group of organisms. It does not represent the evolutionary time or the genetic distance. So this is what a cladogram looks like. You can see all the branches are equal in length. They are not showing any evidence of the time or distance between the organisms. Since branch lengths are not proportional to the number of evolutionary changes, it is also called as an unscaled tree. Phylogram. Phylogram is a branching diagram showing the inferred relationship between various biological species. The distance of branch depends on the amount of inferred evolutionary change. It represents the evolutionary time or genetic distance between the group of organisms. So here you can see the branch lengths differ, which is indicative of the genetic distance or the evolutionary time. Since branch lengths correspond to the evolutionary divergence, it is also called as a scaled tree. Tree topology. The branching pattern in a tree is called tree topology. That can be of two types, dichotomy and polytomy. Dichotomy. When all branches bifurcate on a phylogenetic tree, it is referred to as dichotomy. In this case, each ancestor divides and gives rise to two descendants. So this is a dichotomy. Polytomy. Sometimes a branch point on a phylogenetic tree may have more than two descendants, resulting in a multifurcating node. The phylogeny with multifurcating branches is called polytomy. So this is polytomy. Degree of tree resolution. Star tree. A cladogram that is completely unresolved is termed a star tree. This is a star tree. Partially resolved. A cladogram that has a mixture of dichotomy and polytomies is termed as a partially resolved tree. This is what a partially resolved tree looks like. And a fully resolved tree, a cladogram in which all internal nodes are resolved, that is dichotomy is present and is termed as a fully resolved tree, like this. Types of phylogenetic trees. There are two types of phylogenetic trees, rooted tree and unrooted tree. Unrooted tree. An unrooted phylogenetic tree does not assume knowledge of a common ancestor but only positions the taxa to show their relative relationships. So this is an unrooted tree. We are not getting any information about the common ancestor. There is no direction of an evolutionary path. It is less informative. We don't know how the evolution is taking place and in which direction. So this type of a tree is a little less informative. Rooted tree. All the sequences under study have a common ancestor or root node from which a unique evolutionary path leads to all the other nodes. So this is a rooted tree. So there is a common node or a root node which gives rise to its descendants. The direction of evolutionary path can be traced, and this type of a tree is more informative. Converting unrooted tree to rooted tree. It is often desirable to define the root of a tree. So there are two ways to do that, outgroup approach or midpoint rooting approach. Outgroup approach. Outgroup is a sequence that is homologous to the sequences under consideration but separated from those sequences at an early evolutionary time. Outgroups are generally determined from independent sources of information. So if this is a phylogenetic tree, we can classify them into in-group and out-group. So out-group has diverged very long time back during the course of evolution, and it is evident from this diagram. And in-group is the closely related species. All the sequences are homologous to each other, but out-group has diverged very early in the span of their evolution. For example, a bird sequence can be used as an outgroup for the phylogenetic analysis of mammals based on multiple lines of evidence that indicate that birds branched off prior to all mammalian taxa in the in-group. Outgroups are required to be distinct from the in-group sequences but not too distant from the in-group. Using two divergent sequences as an outgroup can lead to errors in tree construction. Midpoint approach. In the absence of a good outgroup, a tree can be rooted using the midpoint rooting approach, in which the midpoint of the two most divergent groups judged by overall branch length is assigned as the root. This type of rooting assumes that divergence from root to tips for both branches is equal and follows the molecular clock hypothesis. And what is the molecular clock hypothesis? Molecular clock is an assumption by which molecular sequences evolve at constant rates so that the amount of accumulated mutations is proportional to the evolutionary time. Based on this hypothesis, branch lengths on a tree can be used to estimate divergence time. So here we are using the midpoint as the node from which the branches are bifurcating. So this is an unrooted tree. You can see E can be the outgroup amongst A, B, C and D. So when we use the outgroup method, what we do is we define a root node between the outgroup and the ingroup. So this is how we can convert it into a rooted tree. But when we do the midpoint rooting approach, we define the midpoints between the species bifurcating, and then we define a root node for all the species under study. So this is evident from this type of a representation. Why finding a true tree is difficult? The main objective of molecular phylogenetics is to correctly reconstruct the evolutionary history based on the observed sequence divergence between organisms. That means finding a correct tree topology with correct branch lengths. However, the search for a correct tree topology is extremely difficult and computationally demanding. The reason is that the number of potential tree topologies can be enormously large even with a moderate number of taxa. The increase of possible tree topologies follows an exponential function. So, the number of rooted trees, that is NR, for n taxa is determined by the following formula. NR equals to 2n minus 3 factorial divided by 2 to the power n minus 2 into n minus 2 factorial. For unrooted tree, the number of unrooted tree topologies NU can be calculated by NU equals to 2n minus 5 factorial divided by 2 to the power n minus 3 into n minus 3 factorial. So using this, we can actually determine a number of topologies for a given taxa, and then we can see that which one is giving the correct tree or not. So these are all possible tree topologies for three taxa, for unrooted tree and for rooted tree. And in the next, we have for four taxa, for unrooted and rooted tree respectively. This can be calculated with the help of the formula which was defined before. This is an end to this presentation. Thank you. Next, we are going to talk about steps in phylogenetic analysis. If you found this video informative, please like, subscribe and share. Follow us on Instagram and Facebook. And for any queries or suggestions, please write to us at the given email address. Thank you.